Hi, welcome to next lecture about cytology. This lecture will be about cytoplasm and its structures. So once again, protoplasm is divided into cytoplasm that we're going to talk about today, and also karyoplasm. And karyoplasm is basically a nucleus, because karyo means nucleus. So that is everything in the nucleus, and cytoplasm is everything around the nucleus. Cytoplasm is made of semi-liquid matrix or cytosol, and it contains cytoplasmic structures like organelles, cytoplasmic inclusions, cytoskeleton, and we can divide organelles into membranous and non-membranous. Membranous organelles basically are the organelles that have a membrane around. They are permanently present in a cell and they contain enzymes for needs of a cell. We have already talked about mitochondria. The previous lecture was about mitochondria, but there are also other organelles in the cell, like endoplasmic reticulum, Golgi complex, lysosomes, and peroxisomes. So first organelle, endoplasmic reticulum. Remember, it's a membranous organelle, so it has its membrane. And really, endoplasmic reticulum creates a convoluted membranous network. So it's a network which is made of channels that intercommunicate with each other. And these channels are cisternae or tubules, and they are all connected with each other. So yeah, that's really how it works. Everything is connected. And really, we differentiate granular or rough endoplasmic reticulum and agranular or smooth endoplasmic reticulum. And you probably know that granular or rough endoplasmic reticulum is rough because of ribozomes that are attached to the endoplasmic reticulum. So there is some kind of cisternae here, a section through it, and there are ribosomes attached to the membrane of endoplasmic reticulum. So that's why it's rough endoplasmic reticulum. And smooth doesn't have ribosomes, of course. So there is, once again, granular is made of cisternae mostly, and agranular or smooth endoplasmic reticulum, which doesn't contain ribosomes, is mostly made of tubules. And what is important is that there is only one endoplasmic reticulum in each cell. So rough and smooth endoplasmic reticulum are just two forms of one endoplasmic reticulum in the cell. So imagine that these two are just two forms of one thing that is in the cell, and there is only one endoplasmic reticulum in the cell. So ribosomes are attached to the endoplasmic reticulum, but they can be freely in the cell. They don't have to be attached to the endoplasmic reticulum. But ribosome is an integral part of rough endoplasmic reticulum and is described as a non-membranous organelle. So ribosome doesn't have a membrane and is very small, so we're not able to see ribosomes in light microscopy, but we can see them in electron microscope. They are 20 times 30 nanometers big, or small, I should say. They are 20 times 30 nanometers small. So you can see ribosomes only in electron microscope, and we use them as a standard measurement for other structures in the cell. In light microscopy, as you probably remember, we use erythrocytes or red blood cells as a standard measurement for light microscopy, and in electron microscope we use ribosomes. A ribosome is composed of a big subunit, which is this big structure down below here. This is a big subunit, and this is a small subunit on the top, sitting on the top, small subunit. Yeah. 3 molecules of ribosomal RNA, so 3 molecules of ribosomal RNA are in the big one, and 1 molecule of ribosomal RNA is in the small subunit. So together there are 4 molecules of ribosomal RNA that are creating ribosome with another proteins that are in there. Really, ribosomes are also in prokaryotic cells, but they are smaller, lighter. And this is a picture from electron microscope. This is a ribosome. It's hard to see it, but really you can see it. And this is a big subunit here. This is a big subunit, and this is a small subunit. And these are the pictures of separate subunits. So these are the big subunits, and these are the small subunits, but like whatever. No, you don't have to like differentiate the ribosomes in such magnification. So okay, so this is picture from the light microscope, and we can see a neuron here, 2 neurons here. So this is 1 neuron here, and the second one is here, with a quite light nucleus in the middle. So this is 1 nucleus, there is another nucleus of a neuron, and neuron has got some processes. So there is a process here that is going out of the cell. These are the processes that you are going to learn about a lot, so don't worry, we get there. And you can see this violet, or you should now know the term basophilic. We can see basophilic lumps, or I don't know, nodules or something in the cytoplasm of a cell. And really what this is, these are the ribosomes. And now you're like, what? I thought you said we're not able to see a ribosome in a light microscope, and now you're describing ribosomes. What's with that? Yeah, you're right, but when there are too many ribosomes in a cell, they create, in this neuron, they create lumps this big, and you can see them in the light microscope because there are just too many of them. It's not like you would be able to see 1 ribosome that is just alone here, but you can see really a group of ribosomes. So these are the groups of ribosomes, and in neurons we call them Nissl substance. Nissl substance. Nissl substance. So Nissl substance represent an accumulated ribosomes in the cell, basically. And why are they basophilic? Really, ribonucleic acid is an acid. So acid binds hematoxylin, which is a base, and hematoxylin is violet or blue. So that's why ribosomes are basophilic. When there are plenty of ribosomes in the cell or in the cytoplasm, we say that the whole cytoplasm is basophilic. So because it contains a lot of ribosomes, so I would describe the cytoplasm as basophilic. The whole cytoplasm of neuron is basophilic because it contains a lot ribosomes. Why it contains a lot ribosomes? Well, probably this cell is producing a lot proteins, many proteins. In this picture, we can see that rough endoplasmic reticulum, which is very interesting, is connected with nuclear envelope. So here is the nucleus of a cell, and you already know a nucleus has its envelope, which is made of two membranes, and there are also ribosomes attached to the nuclear envelope of a nucleus. So there are these ribosomes also, and this nuclear envelope is interconnected with rough endoplasmic reticulum right here. So this is the rough endoplasmic reticulum. So really also rough endoplasmic reticulum and nuclear envelope are really very close to each other, and they are interconnected, so probably they are the same structure. Can't really tell, but a nuclear envelope is a little bit different, but really on the other hand it's so similar to endoplasmic reticulum. So sometimes also a nuclear envelope can produce proteins also, because it has ribosomes attached to its surface, its membrane. Okay, but how it is really with the free and bounded ribosomes? Free ribosomes produce proteins for the cell. So if you have a free ribosome or free polyribosome, it produces proteins for the cell that are not intended for secretion, or proteins that are not dangerous. These proteins travel to their final place via intracellular transport. These are, for example, nuclear proteins or cytoskeletal proteins, or proteins like hemoglobin in red blood cells. These proteins are for the cell, and these are synthesized on the ribosomes that are not attached to the endoplasmic reticulum. But we have also other proteins like secretory proteins that we want to secrete, or lysosomal enzymes, which are very dangerous for the cell. So you can't have a lysosomal enzyme just floating around in the cell, because it will probably destroy something. So you need some kind of membrane around the enzyme to protect the cell. Or integral membrane proteins. These three types right here that are written right here, secretory proteins, lysosomal enzymes, integral membrane proteins, are very important, and you should remember them, because these proteins are made on the ribosomes that are attached to endoplasmic reticulum, that are attached to its membrane, like this. So, but like, how does the ribosome know where to attach and where to go and what to produce? It's very complicated. Not that much, but it's kind of complicated. You can see it right here. A proteosynthesis begins on the free polyribosomes steadily, always. A polypeptide chain is formed like this. There is a polypeptide chain. Of course, there is a messenger RNA. That is, ribosome is translating this messenger RNA, and it's creating a new polypeptide chain right here. Proteosynthesis starts always on free polyribosomes, always. And this chain, this new polypeptide chain, is called signal chain. Signal chain. When this sequence is formed, the ribosome recognizes it and interrupts proteosynthesis. So it's like that you started the proteosynthesis, and now there is this signal chain, and the ribosome is like, whoa, 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 stop. We need to get to the endoplasmic reticulum, because if we're not going to get there, we're probably going to create some enzyme which is going to be very dangerous for the cell. So it just interrupts the proteosynthesis. So it just created this signal change, and now the proteosynthesis is stopped until it binds to signal recognition particle, SRP. I don't know if it's written somewhere here. I'm going to write it down. Signal recognition particle. That is normally free in cytoplasm, so just signal recognition particle is just flowing around, and the ER membrane, or the endoplasmic reticular membrane, has the SRP receptor, so the receptor for this particle. So ribosome can attach to this receptor with the help of this signal recognition particle, and also it attaches to the ribosomal receptor right here. So also it is attached to the SRP receptor and also ribosomal receptor right here. And we have two types of ribosomal receptors, and one is riboforin 1 and riboforin 2. And riboforin 1 serves as a receptor that ribosome is attached to, and riboforin 2 serves as a channel. Like here, there is this channel through which is the newly synthesized protein chain going through to the membrane. So right now, this new protein change that is made is separated from the cytoplasm. So it's in the endoplasmic reticulum. So it works like this. Yeah, and after that, like when it's used and the new polypeptide chain is made, this signal recognition particle is liberated, and now it can flow around normally, like it flows around. So once again, remember that secretory proteins, lysosomal enzymes, and transmembrane proteins, or integral membrane proteins, are made in the ribosomes that are attached to the endoplasmic reticulum. And there is also this thing called signal peptidase, which really separates the signal sequence. Because signal sequence is just a signal sequence. It's unnecessary residue. You don't have to have a signal sequence in your proteins. It's just for signalizations. So really, what does that mean is that also post-translational modifications begins in the rough endoplasmic reticulum. And I think there is this one question in the test that is asking you, like, where's the start of the post-translational modifications, in which organelle, and the correct answer is endoplasmic reticulum. There's the start. Of course, most of the post-translational modifications are in the Golgi complex, and that's another question. But post-translational modifications start at rough endoplasmic reticulum, and this is it. You can see the one thing here. So when the protein is made, it's stored for some time in endoplasmic reticulum, and transmembrane protein is just moved from ribofluorine to the membrane of endoplasmic reticulum, and then after that it's transported to the membrane of a cell. Okay, so this is a picture from electron microscope, and we can see a cisternae of rough endoplasmic reticulum, because these are the ribosomes attached to the membrane. So cisternae like here, and there is also a nucleus nearby, and that is really okay because endoplasmic reticulum is located closely to the nucleus of a cell. You know that endoplasmic reticulum is interconnected with the nuclear envelope, so this is pretty much okay. If the cell is active in the terms of protein production, there are many cisterns of endoplasmic reticulum, like this. And this cell right here really developed an ability to store its product directly in the endoplasmic reticulum. So that's why we can see a dilated cisternae of rough endoplasmic reticulum. This is really rough endoplasmic reticulum right here. You can see this dilation. It's because really here in cisternae there is a product which is the cell creating. And I can tell you right now, this is a plasma cell. So this is a plasma cell, which is an effector cell of B lymphocytes. So B lymphocytes differentiate into plasma cells, and these plasma cells create or produce immunoglobulins or antibodies like this. And these antibodies are stored in here in endoplasmic reticulum. And we can see also a mitochondria here. That is pretty much normal. Every cell has mitochondria, so that's really okay. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum is made of tubules mostly, and you now know that is interconnected with rough endoplasmic reticulum, and that is only one endoplasmic reticulum in the cell. And there are no ribosomes attached to the smooth endoplasmic reticulum, or maybe just a few of them. That's pretty normal. In the muscle, we can see a combination of tubules, like here, the tubules, and cisternae. So this is the combination of cisternae and tubules in smooth muscle, and not in a smooth muscle, in skeletal muscle. I'm sorry, in skeletal muscle we can find this combination. And why do we have smooth endoplasmic reticulum? This is a picture from electron microscope, and these round oval things, cross sections of the tubules, are really smooth endoplasmic reticulum. So this is the smooth endoplasmic reticulum right here. You can see right now that sometimes we can see also ribosomes that are freely in the cytoplasm or creating polyribosomes, like here there is a polyribosome, and sometimes we can see there is like this cross section of a tubule of smooth endoplasmic reticulum, and there is this black dot sitting there. So sometimes ribosomes are attached to the smooth endoplasmic reticulum, but not so many like in a rough endoplasmic reticulum. And what are the functions of smooth endoplasmic reticulum? Are that it it is a synthesis of phospholipid, so basically the synthesis of membranes, and also steroid synthesis, glycogen storage synthesis, and like this cleavage cleavage of glycogen, and also detoxification. So it can oxidize, conjugate, and do methylation. And really, smooth endoplasmic reticulum has a different enzymatic equipment. It has other functions. So really, for granular endoplasmic reticulum, it was almost only a proteosynthesis and some post-translational modifications of the proteins. For the smooth endoplasmic reticulum, the situation is more complex. It's really for the steroid synthesis, the glycogen cleavage, detoxification of a cell, synthesis of phospholipids, very important functions. Okay, so we have other non-membranous organelles. We started with ribosomes, but we have other non-membranous organelles like proteasomes, chaperones, and vaults or nanocapsules. Well, and proteasomes. Proteasomes are really protein complexes. They are electron dense, and they are 10 times 60 nanometers big. So they are bigger than ribosomes, but thinner than ribosomes, and they are longer, of course. And there is this barrel-shaped model of two lids that can be opened, one on the top and the second one here. And really, they serve to remove proteins. If there is a protein that is set for the degradation, it has to be marked with ubiquitin. So there is this protein that is old and needs to be degraded, and really ubiquitin is a small protein that marked the protein that is set for degradation. And now the proteasomes can recognize that this protein is probably bad or is old, so it digests the protein. And there is also some kind of recyclation of amino acids. So it's not that all protein is destroyed and you're going to get nothing from it. So there is a recyclation of amino acids in the cell. Chaperones. Well, these structures are another non-membranous organelles, and they just serve to form tertiary or quaternary structure of a protein, and that's why they are here. Bolts or nanocapsules. I think you don't have to remember this number, but they are bigger than ribosomes, and they are really just for the transportation of some stuff. Not that important. They are quite large and they transport things. They really look cool, I think. Just I want to be transported in such a thing. I like them, but they are not that important. Golgi complex, on the other hand, is very important. So, Golgi complex is similar to smooth endoplasmic reticulum, but it differs in that its components, like these cisterns, it really looks like smooth endoplasmic, but its cisternae are not connected. They are isolated. So you can find only one Golgi complex in the cell, which is made of several cisternes, like 10 of them, for example, but they are separated from each other. So it's not the interconnected channels like in endoplasmic reticulum. They are just separated cisternes in the Golgi complex. And remember, there is only one Golgi complex and one endoplasmic reticulum in the cell. We distinguish the side cis, or cis phase, which is towards the nucleus. So somewhere here is nucleus. And we differentiate the trans phase. Trans phase has a concave shape, and really Golgi complex emits substances here. So this side, the trans phase, is facing away from the nucleus to the outside world of the cell. Not that it's located outside of the cell, but it's facing away from nucleus, so the opposite side. And really Golgi complex is made of several tanks or several cisternae, like endoplasmic reticulum. You can see it right here. This is the Golgi complex, and you can see flat cisternae right here. And sometimes they are a little bit dilated, but you can see also the vesicles that are around here, and they are also part of a Golgi complex. And we can see also some bigger structures all around, and these are vacuoles, also a part of Golgi complex. There is also a Golgi complex right here, some cisternae right here, and it really looks like there are many. There is one Golgi complex, second Golgi, and third one, but really that's not true. There is only one Golgi complex, which is made of separated cisternae, and you can see it right here in a fluorescence microscope photo, and you can see how big Golgi complex is, because this blue structure right here, that is a nucleus, and this red structure around is a Golgi complex. So really it's really big. So once again, we have a cis phase right here and trans phase right here. And really what's happening is that Golgi complex receives vesicles which contain proteins from endoplasmic reticulum. So these are the transporting vesicles which are going from endoplasmic reticulum to the cis phase of a Golgi complex. And these cisternae in Golgi complex are separated, so there have to be another vesicles around. You can see it right here. They're transferring to another cisternae right here, and they are released in the trans phase right here. And what is Golgi complex doing is that Golgi complex serves as the main seat of post-translational modifications. They start in ER, as I said before, signal peptidase, for example, but the main seat of post-translational modifications are happening in Golgi complex right here. Remember these transport vesicles or transportation between organelles. These transport vesicles are moved or released by a protein called dynamin. Yeah, dynamin is the protein that releases the transport vesicles here. And what are these post-translational modifications? They really are carbohydrate chain modifications, phosphorylation, sulfatation, or proteolysis, so cutting the proteins to some smaller parts. And these are the post-translational modifications. And there is another picture, we can see it right here, which basically tells the same thing. Nothing new here. Next, we are going to talk about lysosomes, which are membranous organelles in the cell. And really, lysosomes are spherical, they're relatively small, they are below the level of light microscopy, but they can be also larger. Sometimes there is one exception from that, and we can observe the so-called azurophilic granules in light microscopy. So the only way we can see lysosomes in light microscopy is to look on azurophilic granules, which are in some cells in immune system. So, but normally you can't see lysosomes in light microscopy, but in electron microscopy, which we have a picture right now, we can see lysosomes. Okay, so lysosomal system has stages of maturation, and the earliest stage of development is the lysosomal transport vesicle. This vesicle is separated from Golgi complex, of course. As I told you before, lysosomal enzymes need to be produced on the ribozomes, which are attached to the endoplasmic reticulum. From endoplasmic reticulum, they are transported via vesicles, which are released via dynamine into the Golgi complex, where are these post-translational modifications for these enzymes. And from Golgi complex, Golgi complex creates these lysosomal transport vesicles, which contain lysosomal enzymes, but they are inactive. So they are not working at this time. Because really what you need for the vesicle in order to activate its enzymes is that you need to lower pH. So you need quite low pH for enzymes to activate. But lysosomal transport vesicle have high pH, so they have inactive enzymes. Normally they are homogeneous, electron dense. We're going to see that. And yeah, when lysosomal transport vesicle fuse with something called endosome, it creates a lysosome. So lysosome is the next stage. Lysosome has got a heterogeneous content. It has got an active enzymes in it because this endosome contains proton pumps, which are lowering pH, because where are protons, there is low pH. So protons are for the low pH. So there is an acidic environment which activates the enzymes, and there really is digestion going on in lysosomes. And gradually the only thing that stays in the lysosome are indigestible substances. So when you can't digest something, it stays in the terminal lysosome. This is the result of digestion. It's called terminal lysosome or residual body, so it's a residue. There is no longer any enzyme activity here, and this terminal lysosome will be excluded. It's called cell defecation. Yeah. Sometimes it doesn't have to be excluded. Sometimes the cell want to keep its poop inside, and that's why sometimes we can see lipofuscin, which is a product of degradation of some pigments and lipids and stuff. So sometimes you can see really terminal lysosomes, which are in big amounts located in the cytoplasm, a part of cytoplasm, and you can see them, and they are in the long-living cells like neurons or cardiomyocytes and these cells. So these cells are keeping their poop. Don't tell this on exam, please. So lipofuscin, once again, is a lipid degradation product. It's a yellow-brown pigment which we can sometimes see in the long-living cells, because there are these terminal lysosomes together and located together in this part of cell in the cytoplasm. So how does the lysosome form, or how does the Golgi complex create lysosomal transport vesicle? Is that enzyme, this, for example, lysosomal hydrolase precursor, is labeled with something called mannose 6-phosphate. So mannose 6-phosphate is like a label. It labeled enzyme, and really it's very unique. It's a unique label, so you should remember this mannose 6-phosphate. And really this enzyme with this mannose 6-phosphate is already going through all the Golgi complex right here to the trans phase of Golgi complex, where it is released, because there is a receptor for this mannose 6-phosphate. It's called mannose 6-phosphate receptor. It's quite nice. And really this lysosomal enzyme is captured here, and the piece of membrane is separated right here from the Golgi complex, and the lysosomal transport vesicle is formed like this. So really the Golgi complex gives its membrane to create a vesicle that contains this enzyme, which is bound to a receptor because it was labeled with mannose 6-phosphate. And still, remember, this enzyme right here is in lysosomal transport vesicle. There is a high pH here, so this enzyme is inactivated. And lysosomal transport vesicle looks like this. Really homogeneously electron dense particle in the cytoplasm of some cell. So very, very high electron density and very homogeneous lysosomal transport vesicle. There is a whole mixture of enzymes here. Not just that hydrolase, but also acidic phosphatase, ribonuclease, and all other stuff. Here, the yellow color here, acidic phosphatase, is always there, and serves as a proof of a lysosomal presence. So really, what does acidic phosphatase do? It cleaves the phosphate from its substrate, but this acidic phosphatase can be very well detected. So that's why we use it as a proof of lysosomal enzymes in the cells. So there is always some intake on the cell surface, and ingested substances are in vesicles. So you remember pinocytosis, phagocytosis, or receptor-mediated endocytosis. So for example, here we have a pinocytosis. There is some substance here that the cell wants to eat, and it creates, through the process of invagination of its membrane, it creates a pinocytic vesicle, PV, right here, there, that has substrate inside. And also these pinocytic vesicles fuse with vesicles like here, for example, like here, that were also created through the process of invagination of a membrane. And these vesicles right here have a proton pumps in its membrane. So a lot of proton pumps. That means there are many protons in this vesicle. That means that this vesicle has a low pH on the inside, so it's acidic. This vesicle is very acidic. So when the pinocytic vesicle fuse with this proton pump vesicle, I don't know the name for this vesicle, but it's not important, it creates something called early endosome. So really, early endosome is a structure that contains a substrate from pinocytic vesicle and also that have a low pH. So it has a substrate and also low pH. This early endosome is transported along microtubule deeper to the cell, and this transportation, or this vesicle that is transported, is called endosomal transport vesicle. So that's like next stage of an early endosome. We have an early endosome and it's going deeper along microtubule. Then we call it endosomal transport vesicle or multivesicular body. And when it's deep enough, we call it late endosome because it's older. It's like late endosome. I'm sorry. And when late endosome fuses with lysosomal transport vesicle, which was created by Golgi complex, lysosomal transport vesicle, or I should say rather, which was released by Golgi complex, but because it was created in endoplasmic reticulum, and then they were some kind of post-translational modification is Golgi complex, and then Golgi complex released this lysosomal transport vesicle. So when the multivesicular body or this endosomal transport vesicle creates the late endosome, and this late endosome fuses with lysosomal transport vesicle, it creates a lysosome finally. So that's how we get from lysosomal transport vesicle to lysosome, is that now we have low pH also here in lysosome because of the low pH in the endosome, and we have the substances that we need really from the pinocytosis, for example, and we also have the enzymes. We also have the enzymes from the lysosomal transport vesicle. Okay, so these are the pictures from the lecture before, and these are the pinocytic vesicles right here. Pinocytic vesicles in a cell that is transporting. And these are the receptor mediated. This is the receptor mediated endocytosis, and this is so we call this structure a coated pit, and coated vesicle right here, because it has coating proteins. Like you remember, clathrin is a coating proteins, but there are many others, but clathrin is the most important to remember. So clathrin proteins. So pinocytic vesicles, they randomly receive molecules from the environment. So they're just invaginations of the membrane, and you're creating pinocytic vesicles randomly, picking up everything around, not everything, but most of the stuff around. And this receptor-mediated endocytosis is very specific. So you have a receptor for something, and then you just creating a bit, you invaginate the membrane. So this is just randomly picking up substances, and this is very receptor-mediated stuff going on. So very selective transport. Okay, we can digest also organelles which are in the cells, and we call these particles autophagosomes. This is the tubular type of mitochondria. You should probably remember that tubular type of mitochondria is located in the steroid producing cells, and I can tell you right now that steroid producing cells have mitochondria, tubular, and also steroid producing cells have a lot, or have a big smooth endoplasmic reticulum. Smooth endoplasmic reticulum because smooth endoplasmic reticulum contains enzymes for steroid production, and also steroid producing cells have also big amount of lipid droplets because they need the lipids for the steroid production. And how did I get there? Like from the autophagosome of mitochondria that is tubular. Very well. But okay. So you can digest your own organelles. And heterophagosome is a structure that's created from a process of phagocytosis. So when you fuse endosome with the phagocytic phagosome, it's called heterophagosome. This is an endosomal transport vesicle which is transported along microtubule right here. So it's a part of endosomal system, and it's called multivesicular body because there are a lot of vesicles on the inside, but they just look like vesicles. Normally this cell, not this cell, I'm just talking shit right now. This is not the cell, this is just the endosomal transport vesicle. This vesicle, its membrane is creating invaginations. So this vesicle looks like something like this, probably. Not like this, but never mind. And when you do a cross section through this structure, you would probably see a round circle with vesicles inside, but these really aren't some vesicles like in here. These aren't vesicles, but these are just invaginations of a membrane. So some of you might ask, what are these vesicles for? So I'm explaining, these are just invaginations. So there is just one vesicle. There's no such thing like as a vesicle in the vesicle, and why are there small vesicles in the big vesicle? So no, they are just invaginations of a membrane. And this multivesicular body is transported and then creates something called late endosome, which fuses with lysosomal transport vesicle and create a lysosome. This schema is really not so correct, I think, because normally there should be this arrow, like we have pinocytic vesicle that fuses with some proton pump vesicles which have protons inside, and when they fuse together they create something called early endosome, and this early endosome is transported like endosomal transport vesicle deeper to the cell, creating late endosome which fuses with lysosomal transport vesicle from Golgi complex and creates a lysosome. And you can see here the explanation of autophagosome. There is a mitochondria on the inside that fuses with early endosome, and there is this process going on. Once again, it creates a late endosome and fuses with lysosome. Heterophagosome, the same. These are the lysosomes. I think you remember lysosomal transport vesicles. There were these dense, electron-dense structures, homogeneous, but these are more heterogeneous and bigger. So they are not that dense as they were lysosomal transport vesicles. So they are lighter. So these are early lysosomes, and these are late lysosomes, because we can see these funny structures right here, like some vortexes in the lysosomes. And what it really means is that the digestion has come to an end. These are the late lysosomes, so this is the ending of digestion. But you can still find enzymes in these late lysosomes. And these are the terminal lysosomes. Here is the nucleus, and this is a terminal lysosome, and also here. So these are the terminal lysosomes. You can see some substrate on the inside, but there are no enzymes in there, and because the digestion just ends. And really the cell can exclude these terminal lysosomes in the process of cell defecation. And as I told you before, some cells can keep their poop inside, and the accumulation of terminal lysosomes can sometimes be seen, like a lipofuscin in neurons or cardiomyocytes. Like this. So here we have this brown pigment, lipofuscin, this brown pigment, brown-yellow pigment, which is in the neurons. So these are the neurons. These are the neurons. And these cells live very long. So that's why we can see this accumulation of terminal lysosomes here. So lipofuscin is a degradation product of lipids. Another organelles are peroxisomes. Peroxisomes are least studied organelles, and they are developmentally very old. These organelles are where oxidative metabolism took place even before they were mitochondria, so they are very old, and they really resemble lysosomes. We can't find acid phosphatase in them really, but we find oxidases there, like catalase, which serves for the degradation of peroxide here. And peroxide is toxic to the cell, so that's why you need to have a catalase to degrade the peroxide, because peroxide is really a product of metabolism that is normally created in the cells. And this catalase, which degrade hydrogen peroxide, can be proved. So it is a marker, like acid phosphatase for lysosomes. So we have catalase for peroxisomes. And really, oxidation takes place in peroxisomes, so they are very important for beta oxidation of very long-chain fatty acids. And this is very important because there are some peroxisomal disorders which affect nervous central nervous system, because if you can't degrade very long chain fatty acids, they accumulate and they destroy nerve endings and neurons. So this is very important from the clinical point of view. So remember, beta oxidation happens in peroxisomes. And of course their content is transported from polyribosomes using address sequences. Everything is using address sequences. And this is how they look like. This is a picture from electron microscope. So really you can't tell difference from lysosomes. Really, they look like lysosomes or lysosomal transport vesicles. Sometimes they look like it. And really there is a picture, sometimes you can see some kind of content on the inside called nucleoid, so, but not in humans, I guess. There is some crystals of uric acid on the inside. So, when they grow up, they split up. When they are big, they just fission into two separate daughter peroxisomes. So they can divide. That's what also mitochondria can do. And the last thing we are going to talk about is cytoplasmic inclusions. Cytoplasmic inclusions, they are very unstable structures. They are temporary, so really they do not have to be in the cell at all. There also may or may not be a membrane around, so some inclusions have membrane, some do not. And they don't contain enzymes, but if they do, so normally they don't contain enzymes, and if they do, they are not engaged in the cell metabolism. So it's just enzymes for excretion or something. And so we can distinguish pigment inclusions here. This is a big group, pigment inclusions, and cytoplasmic deposits. So pigment inclusions and cytoplasmic deposits, two big groups. Pigments have their natural color either by themselves, so they can be like endogenous pigments or exogenous pigments. We can have a pigment from a blood. So when it's from blood, it's called, when it's from blood, it's called hematogenous. So hemato means like from the blood. Or autogenous, like for example, already mentioned lipofuscin. Or melanin. Melanin is also a pigment in the cells. Cytoplasmatic deposits can be divided into lipids, saccharide, and proteins. So let's have a look on some of these inclusions. For example, lipid droplets. Here we have two pictures. One is from light microscopy and one is from electron microscopy. We can see right here there is a mitochondria that is tubular. So once again, this is a steroid producing cell because it contains tubular type of mitochondria, and also we can see a large lipid droplet here. So here this is the lipid droplet, and also here is another one. So this is the lipid droplet also, and this is the mitochondria right here. And as you can see, I need to erase these parts. As you can see, there is no membrane around the lipid droplets. So lipid droplets most of the time don't have a membrane, there are just some cytoskeletal proteins around. For example, vimentin is sometimes around here. vehementin. But there's no need for a membrane around the lipid droplets. And here we have lipid droplets here in light microscopy. These are these blank spaces. So really there is just space here, free space, because really the contents have dissolved, so they are really empty. These lipid droplets, there is no lipid here, there is just blank space here, because we used a method that like flash away the lipids in the cells. So really there is just blank space, but there were lipid droplets right here in these cells, and these cells, this whole cell right here, is called adipocyte. So adipocytes have a lot of lipid droplets on the inside. So really in a picture of light microscopy, the contents have dissolved, so they're just empty. We can see only empty lipid droplets in light microscopy. But there are many different ways how to stain a slide. So sometimes we can see lipid droplets, like here. These are these dark blue things over here. These are the lipid droplets stained with toluidine blue or oil red right here, but that's not that important. But it can be stained. Lipid droplets can be stained. So remember, first inclusions, cytoplasmatic deposits, are lipid droplets, and we can see them in electron microscopy like this, light or lucent, electron lucent structures in a cell, or in light microscopy these small empty spaces because they dissolved. And the next inclusions are glycogen particles. Next inclusions are glycogen particles, which are really electron dense particles. And glycogen is a polysaccharide, so the main storage of saccharide is glycogen. And really these particles are smaller, they're below the level of light microscope, but they clump together. And these first particles, glycogen particles, are called beta particles. So these really small particles are called beta, or like beta. Yeah, particles. And when they clump together, they create something called alpha particles. So alpha particles are bigger. Alpha particles. These are alpha particles, and they are below the level of light microscope, but when they clump together, they accumulate in one part of the cytoplasm. They really can be seen in light microscopy. And this is a very good picture because it's like, I don't like it, but it's stained with carmine, which is something that I'd never used before, but yeah, it's stained glycogen in the cells. So the glycogen here are these very red parts of the cells. But more often on the practical trainings we use a reaction called PAS. This reaction is called periodic acid shift, and it's very useful to visualize glycogen particles. So we will use PAS most of the time, not carmine, but you can remember also carmine. And proteins. Also, there are some protein inclusions, and these are the secretory granules, for example. So the first type of protein inclusions, yeah, so this is the protein, really, are secretory granules here. And we can differentiate serous secretory granules and mucous secretory granules. These granules can be eosinophilic or they can be basophilic. So these are the pictures from light microscopy, and we can see these dots, very basophilic dots. These are the serous granules. So we can see them like dots in light microscopy. They could be eosinophilic or basophilic, and in electron microscopy we can see them as electron dense structures which have a membrane around. So there, in this type, there is the membrane around these secretory granules, and these are really serous secretory granules. They have watery content, and after excretion from the cell, it is a liquid. They contain, these secretory serous granules contain a large amount of protein. There are often digestive enzymes of the pancreas, of the salivary glands. So there is a large amount of protein in these granules, a pure protein, I guess. On the other hand, there are mucous granules, which are very much different from the serous granules, as you can tell. Serous granules are smaller, there is a membrane around, they are separated from each other, not touching, only a little bit touching, but not fusing at all. And mucous secretory granules are opposite. They're light, so they're electron-lucent, very electron-lucent, and they fuse together. They really fuse together. They have very dense secretions, which forms on the surface, and it's called mucus. So there is a mucus on the inside of these granules. Really, in light microscopy, they are visible, but they're not very well stained. So we called this they are optically empty. Like there's nothing in there, but there is. We can see it, but it's not very much stained very well. But these are the mucous granules here. And remember, they are bigger, they fuse together, they contain mucus, and they are electron-lucent. And we call, or we describe these granules, that this material in the granules, as fibrogranular material. Yeah, so we describe granules in electron microscopy here. These mucous granules are fibrogranular. They have fibrogranular material on the inside. And in the light microscopy, we described this structure as a honeycomb. It's a honeycomb structure, or honeycomb-like appearance. It looks like honeycomb. So this mucus, on the other hand, has got glycosylated proteins. It's made of glycosylated proteins, not like serous. And all these granules are surrounded by membrane. There are some other secretory granules, like for example dense core vesicles, like here. They're very special because they have a dense core on the inside, or this lamellar body right here. These are very special types of secretory granules, or specific granules, like that granules that contain insulin, like this. They look very strange. And also proteins can be stored without membrane, like for example crystalloids. These are the example of inclusions. They were made of proteins. They do not have a membrane around. For example, these proteins, these crystalloids. And really, we do not know what they are for, but they are in the cells, which you are going to learn about. And they're very typical for them, but we do not know why they're there. And last but not least, pigment inclusions. So we already said that they are exogenous from the outside world and endogenous. And endogenous are divided into hematogenous, which are made from the blood pigment, and autogenous, which are made in the cells, like melanin. This, for example, this is a melanin. Right here, this yellow brown dot here. Or lipofuscin, already mentioned many times. So lipofuscin here also. We have here hemosiderin. Hemosiderin is a hematogenous pigment, the result of blood pigment metabolism. And sometimes we can find hemosiderin somewhere else, like in lungs, and there is a pathological phenomenon, for example. So you're going to describe hemosiderin inclusions in the lungs, in the cells in the lungs, and that is pathological. So that's why we are learning this. This is very important. And you can see them also through Pearl's reaction. So these are the hemosiderin inclusions, these dark dots here. And also dust particles. These are example of exogenous inclusions, these dark particles in macrophages. Because normally in lungs you have macrophages, or the cells which are trying to clean the lungs, and they eat all the dust particles, and after they do so, they are called coniophages. So these are the, I'm going to write it down, this is the last thing I will do. Macrophages are the same as coniophages in the lungs, but coniophages eaten up dust particles. So these are the cells, coniophages, which are full of dust particles because they're cleaning your lungs. You should be happy that you're having those guys. So I think that's all for today. I hope you understood everything. Once again, I'm sorry I'm sometimes struggling with explanation, but I think I explained everything clearly. If you want to ask something, write down below in the comment section, and have a nice day, and